Hello, welcome back to another mini science session. Does my mic work? Let me just check. Okay, it does. So right now we are going to try hello Apu cheese and cheeseburger. Yes, thank you for joining the membership. Whoop whoop, more emotes. Let's go. Wait, let me type the what's this called? I will type the sparkly. There you go. Okay, yes. Alright, so now we are actually going to try from the very beginning central dogma, from DNA to RNA to protein, and what happens to it after it becomes the protein. So I don't know how long this will take, but we are going to try. And let me just check a couple of things. Yep, there you go. So we know that let's write a thing first. Move. Okay, first central dogma, okay? And the cell journey. So in every living cell, we know that the flow of the genetic info is the foundation to life. We know that one. And this process, aka the central dogma, operates around the same area as the cell cycle. And the cell cycle is just the steps, the sequence of stages that will be for cellular growth, replication, and division. But if we want to, before we go and discuss the central dogma, we have to start with the structure and function of our genome, okay? So this is how it looks like. You have the you have the DNA. This is sugar. And this is the base. And you have the OH here. DNA is deoxyribonucleic acid. Meanwhile, RNA would have something here at the second carbon. So right now we're drawing a DNA. Oh, this is a lot of phosphate. One O, another P, O minus, O minus, 1, 2, 3. Okay, so that's one. And then if you want to connect it to the other one, just draw a second one here. OH. I'll write the bases later. Another O, P, O, and P. 3. Hello. Thank you for coming by, Elmer. This is O, P, O. Alright, that one's finished. And there you go. Now, let's try to connect it from this 1, 2, 3, the third oxygen. Yes, O. Just remember to put the O minus on the phosphates. Oxygen, phosphates, and their charges. There, that's the last one. Connect this to the first phosphate, and like that. You attack that, and then, wait, it would be easier if I just write it this way. Like that, and then there, you cut that off. So now the final product looks like this. You have oxygen, you have OH, you have the base. Again, I will write the base later. O and P, and then like that. This is already connected. Hello, Nguyen, welcome. Thank you for coming by. And then we have the oxygen. I erased it because we're trying to connect it here. O, and then the phosphate, it's now connected here. And then another oxygen before the CH2, and then O, and then an OH and a base. Perfect. And then how this will look like if it's DNA, it can have 4 types of bases. So let's erase them, because for the base we have ATCG, ATCG, ATCG. No, I'll write ATGC, because that's gonna be the order. I will write it in. I will draw them in a moment. So for adenine, it looks like this. No, oh yeah. Nitrogen, not nitrogen here. Line, double bond, nitrogen, R. This looks ugly. Again, 5 first. 1, 2, 3, 4, 5, and 6 over here. 1, 2, 3, 4, 5, 6. Nitrogen, double bond, R. 1, 2, 3, N, NHH. The way I wrote that is important because it will hydrogen bond to what? To thymine. Okay, so hydrogen bond here. Thymine is only one ring, so that's why it's like that. And here, oh, this one should be erased. I want it to connect properly. That's the wrong side. There. This H is connected. Is double bonded. I mean hydrogen bonded to that oxygen. Then you have N, H, hydrogen bond here. And then next is 1, 2, 3, 1, 2, 3, 4 here. 1, 2, 3 here. 1, 2, 3, 4. Perfect. And then NR, double bond O. So that is adenine to thymine. Yes. Now you have guanine to cytosine. It looks like similar to that, except you just have a couple of different things. Oh no, ugly. Draw it again. 6 or 5 pentagon first before the hexagon. Pentagon. Nitrogen. So same, almost the same starting point. Except this is a double bonded O here. Double bonded oxygen. Nitrogen H. NH. Line here. A bond here. And okay. And hydrogen bond. So with that one, you have 6. 1. No. We will do this structure. Yes, 6. This one is double bonded oxygen. N. Oh no, not a pentagon. We needed a hexagon. So draw it this way. Beautiful. And now oxygen, nitrogen, bond, and then nitrogen H. That's where you're bonding. And NR. See? Boom. 3 bonds and 2 bonds. There's a reason why this is more common in like the DNA replication, because it's easier to break 2 bonds compared to 3 bonds. Alright? So if we want to compare, that is, this is DNA, right? Meanwhile, ATP, it looks a bit similar to that, except ATP is using a ribonucleic, like a ribose. It has an OH. So if we want to write ATP versus DNA, draw ATP looks like this. ATP, OH, bond, was it like this? Yes. 1, 2, 3. N, bond, N. This is like this. Here, NH bond N and nitrogen, and then the combination of O P O P O P O, double bond O, oops, no, there, double bond, oxygen minus oxygen minus, what is this, phosphate, oxygen minus. In terms of which ones will break first, you have alpha, beta, and gamma. Does gamma look like this? I think gamma. You break this part first, this one, and so that's how you make it from ATP into ADP. Guess what? T is tri, D is di, adenosine triphosphate and adenosine diphosphate, 2 phosphates. So how that will look like is just two oxygens here, because using a ribose, we have a base, O P O P O, double bond O, oxygen, perfect. See, that is ATP. So the formula breaking it up is ATP plus H2O, you need water to break it down, it's a dehydration reaction, into ADP plus PI plus energy. It's harder if you want to go back to ATP, you need energy to make it. It's easier to just break it up into the smaller form of it. Right, so the DNA, that's a very common word we always hear. So that one, it wraps around histone octamers, okay? So let's write it, let's draw it. Let me just choose the right pen. Okay, it's here. Eraser, I mean. DNA will wrap around histone octamers. So you have this, right? The major groove, the minor groove, major, and minor. Oh, I think I have to make it a little bit thicker. Much better. Okay. Again. So you have this, and the squiggles. You have the major and minor groove. Now, if you just expand that a bit, it will look like this. And this is where you have the A and T R, G to 3C. And then you wrap that around something called a histone octamer. It's octamer because it's like 8 subunits. 1, 2, 3, 4, and the other side. That's 8. If you wrap it around here, yeah. And now it's called a nucleosome. And I'll just write an arrow here. That's the histone octamer. Okay, so that's the first level of the wrapping. And then it will continue to wrap until you have the chromatin. And then until you have something like this. Looks familiar? Yes, that is the chromosome, the X ones. Also, you have a couple of like enhancers, silencers, promoters, and genes. The gene is just a specific segment in a DNA. Yes, Apu, that is the chromosome. That's how you go from this squiggly DNA. You wrap until you get this. And then when people talk about like genes, that's just a specific section of this whole long line. And for example, you have genes for your eyes and all that. And then genes would, for example, need you have the parts of it would be called the enhancer or silencer, and some protein, like regulatory factors, could bind to this one. Let's say it binds to the enhancer, and it will talk to something called the promoter, and then it will promote transcription of the gene. So it will promote the thing it will make. But if the incoming thing will bind to the silencer, then it talks to the promoter, and then no transcription will happen. So that's how things are being controlled inside your body. Okay, so now the expression of genetic information is coupled with the cell cycle. We talked about that earlier. It is a recurring series of stages that prepares the cells to divide. Alright? And in terms of division, you have something called the interphase, such as G1, S, and G2, and then you have the M phase, the mitotic phase. So it's a cycle. So let's draw a circle, A plus circle. Start from the center, and I'll draw something here. I'll fill that up later. So now let's start first with here. You have a cell with some DNA inside, and this one is G1. That's G1 phase. It will continue, and in the G1 you have the cells grow and just do their normal function until, and most transcription and translation happens here. And translation, after it grows, now you can see like, oh, it's okay. It will then go and do synthesis, the synthesis phase. What is here? It will duplicate, if the environment is favorable, it will now duplicate the genetic material inside. So you might have something that looks like an X, because it's double now. And then you have G2, further prepping for division before you go into the M phase, aka the prophase, PMM, AT, and then you have the cytokinesis at the end, because you have interface. This is the whole interface, I, and that before you go to the mitosis part. So M phase, you have the prophase, prometaphase, this is double, yeah, prometaphase, metaphase, anaphase, telophase, and cytokinesis. So in the prophase, it will break down I know that the nuclear envelope will break down, start to break down. Prometaphase, it's finished breaking down. Metaphase, it will all line up in the center. Anaphase, they will split up. Telophase, it's a little bit of the start of the cleavage, and then cytokinesis is when they will fully split into two cells. So that's why you have this and this, two of them. But this one we're just not drawing here because the starting point is just one. Okay, in terms of gene expression, in G1, the transcription factors like the E2F, they are activated to drive the expression of genes through the S phase. But in mitosis, transcription is halted because of chromatin condensation. If the chromatin earlier is too like bunched up, this is a heterochromatin, it's too condensed, you can't have factors coming in because it's too tight. Meanwhile, if it's a euchromatin, then it's a little bit, you can put in some factors there that will help the cell cycle move forward. Okay, now we said transcription a gazillion times before, so let's talk about it. So the whole central dogma, DNA, RNA to protein, it begins with transcription. Okay, the synthesis of RNA from DNA. Okay. And this is inside the nucleus. So if you have the cell here, and this is the nucleus, DNA to RNA, that is inside the nucleus. And the important enzyme is something called RNA Pol 2. The enzyme for transcription, again, what is it? RNA Pol 2. And you can't, by the way, RNA Pol 2 cannot initiate transcription on its own. It requires like a team of a gazillion general transcription factors and regulatory proteins. So how does it start? You have TF2D. It will recognize the core promoter. Okay, this one. It will recognize, you have the Tata box. The TF2D will recognize the core promoter region of the gene, aka the Tata box. And it has the, take note, it has the TBP. What is that? Tata binding protein. It will bend the DNA and form the pre-initiation complex. And then after the TF2D with TBP comes in, you have the TF2A and the TF2B. What do they do? Okay, they would bind the TBP and DNA pair up or interaction and position it correctly. So that's the job of these twins. Now you have, after that one, you have TF2E and TF2H. Those are recruited. What does that do? TF2H has helicase activity. It will split the DNA. It will unwind it. Remember, it looks like a squiggly line like that. We have to split them up. So TF2H has helicase activity and kinase activity as well. So the kinase activity will phosphorylate the C terminal of the RNA Pol 2. I think visually this is the C terminal of it, of RNA Pol 2. So after you add a phosphate here, it's almost activated now. And once initiated, RNA Pol 2 will move along the DNA in 3 to 5 prime direction, and it will make the new RNA in 5 prime to 3 prime. So again, let's make sure we understand that part. Okay, again, write it. If this is 3 prime to 5 prime, it will sit here. This is the DNA, right? It will go in this direction because the growing strand will be 5 prime to 3 prime. That's what it means. And after that, just during elongation, the SIR5, the CTD, remember the C terminal domain, it will just increasingly become modified, and the serine 5 will get replaced with serine 2, and that is when splicing factors and the 3' end processing machinery comes in. But that's after this. Transcription bubble is just maintained as it reads it, and eventually it reaches the end. The RNA pol reaches the end, and then there's no set termination sequence. It will just continue until there is a polyadenylation signal. The poly A, that's why you have to add the poly A tail later. After you have the signal, the A, the RNA is then cleaved, and then it is polyadenylated. Exonuclease will be the one who will help with the endpoint. Alright, now RNA processing, that's the next one, because this is just pre-mRNA. So it still needs to be processed into RNA, proper mRNA. How? Why? What? So let's erase it and figure that out. Okay, so the first thing that happens is the 5' cap. Next is splicing, and then you have poly A tail. Alright, for the 5' cap, almost immediately after transcription, you immediately add, like, a guanylyl transferase will immediately add the 7-methyl G-guanosine cap. 7-methyl guanosine cap through a 5' linkage. But the purpose of it is just to protect the RNA from degradation. And then splicing, you have a couple of factors, U1, U2, U4, U5, U6. Don't ask where U3 is, it's doing something else. But the focus is just U1, 2, 4, 5, 6. And there, U1 would bind to the 5' splice site. U2 would bind to the branch point. 4, 5, 6 will form the catalytic core. And then you just have two trans-esterification reactions, remove the introns. Okay. So why do we want splicing? It's because, for example, you have a couple of exon 1, intron 1, exon 2, and then exon 3. It depends if you just want to keep, first of all, you remove all the introns. We don't want introns here. But it depends if you combine exon 1, exon 2, and exon 3 together, or you could actually make something like just exon 1 and exon 2, or just exon 2 and exon 3. It will depend. And though these mRNAs will act differently, it depends on what kind of protein they can make. So that is why you want splicing as well. And now for the poly A tail, we mentioned it earlier. You just bind to the polyadenylation signal to cut the RNA, and then you add the poly A tail to stabilize the mRNA and also assist in translation. Okay? And to make sure that only properly processed RNAs are translated, cells will employ RNA quality control mechanisms because you want them to be properly controlled. And we will eliminate them via the NMD, the nonsense mediated decay, just so that we don't have a lot of RNA that has premature stop codons. Alright. Oh, and you have the nuclear pore complex. It will only export properly capped, spliced, and polyadenylated mRNAs. In terms of exporting, let me just draw what's the next part. So far, here, erase. We talked about this. It happens in the nucleus. You have DNA transcribing into RNA, and then RNA processing. This is now the proper RNA, and then it has to be transported outside so that you can make proteins. So this process, you have the nuclear, it goes through nuclear pore complex, and only the RNA that are properly capped, 5' capped splicing, and properly poly A tailed are allowed to go out. And if they are not allowed to go out, they will stay inside and be destroyed. Okay. Next is translation. So once in the cytoplasm, the mRNA will undergo translation, and with that one you have different things. You have the ribosomes and the rough ER and stuff like that, but we're just going to focus now on the exact process. So first you have your mRNA. Let me just draw it out 5 prime to 3 prime mRNA. And then first is you have the small ribosome subunit 40S binds to the 5 prime cap. Okay, what that means is 40S will bind to the 5 prime cap. This one, oops, 5 prime cap. Oh, I can just draw it there. 40S. And then, oh, that's done via the 40, the ELF4E. Now, you have the tRNA. It's escorted by the ELF2 GTP to the P site of the 40S subunit. P site would be the one in the middle, because you have the APE. So here, this will have the MET. And now the 40S subunit will scan from 5 prime to 3 prime. It'll go like this until it finds the AUG. And once AUG is formed, GTP will be, once AUG is found, GTP will be hydrolyzed, and you just have, and then you will have the large 60S subunit come in. Hello Joyers, welcome back. Yeah, you have the 60S subunit come in, and it will join to form the 80S ribosome. Okay, that one's so far. And then the next step will be just the 3 major steps: elongation, translocation, and termination. Okay, and remember you have the A site, P site, and E site. For A, the new ones will come in here, and then wait, the new ones will come in here. The amino acid acyl tRNA will enter the A site. It will, anticodon should match the bottom, and after that one, peptidyl transferase will move the one from here to over here, and then all of it will shift. Right now, both of them are at P and A site. This ribosome will move over to this direction, and now both of them will go and be at the EP site, and then that's how the cycle continues until you hit a stop codon, aka the OAG, OGA, and UAA. It will stop and then translocate. The entire ribosome will translocate. Just let go. So that is DNA to RNA to protein. And then now we have protein trafficking and sorting. It really depends on where it was translated. Okay, so if you have the whole cell, this is a big cell, but trust, it will help. So you have DNA to RNA, and then processing, and then you transfer it. You have the mRNA, right? So if that mRNA goes to a free cytosolic ribosome, and then that's how it is made as a protein, then it won't need any vesicles. It will be non-vesicle mediated. Non-vesicle mediated versus if it is inside a membrane-bound ribosome, like at the rough ER or ER, then it will need vesicles. It will be vesicle mediated. So for the free cytosolic one, it will just go to a cytosolic ribosome. For signals, you have 3 here. If it has the NLS, the nuclear localization signal, importin it's recognized by the importin, and after that it will be translocated to the nucleus. But if it has the N-terminal signal N-terminal amphipathic alpha helix, then it goes to the mitochondria. And then if it has the PTS1 signal, it'll go to the peroxisomes. Again, it DNA RNA to protein, and then if that protein was made in the cytosolic stuff, if it has the NLS, it will get transported to the nucleus. If it has the amphipathic one, it will mitochondria, PTS1 to peroxisomes. And then you have the membrane-bound ribosomes now. So it will be the N-terminal signal peptide NSP. And then it will first go to the Golgi before being distributed to, like, as a secreted protein, plasma membrane, and endomembrane system, like the ER, Golgi, and lysosome. Alright. Good. Earlier, talked about the ER. I just want to mention that a bit more. Here. Okay, so in contrast, proteins for secretion or organelles, they are synthesized in the rough ER. They would have the N-terminal signal peptide. It's recognized by the SRP, signal recognition particle. And so the way what that looks like is you have 5', 3'. It is made here, and the new protein has the SRP. And then you have the ER here, ER lumen. And I will just draw it like that. SRP will go to the SRP receptor. And that's how it starts. And then it will dock onto the S61 translocon using GTP. GTP will transfer the signal to the Sex61, and then you hydrolyze and finish it up. And the peptide is released into the lumen of the ER. But there's something called N-linked glycosylation. Because as you enter the ER, you need to protect it as well. So N-linked glycosylation is initiated co-translationally, and it's as a lipid-linked oligosaccharide. So the way N-linked glycosylation works is, let me just draw the membrane of the ER like this. So if you have the glycan here, you have something called the oligosaccharyl transferase, which will just tag it on the nitrogen sites, aka if it's an asparagine, that's why it's N. If it's an asparagine, you have CHCOO minus, H3N plus, nitrogen. Asparagine looks like CH2, C, double bond, no, not double bond O there, NH2, and double bond O over here. So this is where the N is, and if it is co-translational, it's because the oligosaccharyl transferase is OSTA, aka it has the STT3A part of it, and that is how you N-link glycosylate things. And once the proteins are correctly folded and modified, they are actually packaged inside the Golgi apparatus. We always hear that the Golgi apparatus is like Amazon, it'll package stuff and deliver. That's because it is. And this is your ER, right? This is the ER. And if this is the Golgi, squiggles, Golgi apparatus, going from the ER to Golgi, you use the Kpop2. But going back, use Kpop1. Okay? Go again. Kpop2 is going to Golgi apparatus, Kpop1 going back. So it traffic to the cis Golgi network will be, it's via snare mediated membrane fusion, and in the Golgi proteins are processed, and that is where O-linked glycosylation happens. You attach the sugar to serine. Serine could look like serine or threonine. Serine is CHCOO minus H3, and plus serine is CH2OH. Meanwhile, threonine looks like this, H3N plus. This has 3 spots, COH. I think it's this, CH2. Yeah, CH3, just so there's 4 bonds. So that is how things happen. ER to Golgi, COP2, Copite, intra-Golgi, and also going back to the ER, that is COP1. And if it is going to Golgi to ribosome, that is clathrin. That's where clathrin comes in. And then final delivery, proteins embedded in the plasma membrane are, I can just keep it here, are inserted via the transfer signal during ER translocon. Meanwhile, soluble proteins are released outside the cell. And altogether, you have the protein localization. It's very tightly regulated. So yes, that is how you go from the central dogma, DNA, RNA, protein, and then protein trafficking. I think that ends the story for now. And that is all for this mini session. Thank you for stopping by, and have a nice day.